Test mic, test mic. Okay, I think my mic is okay. Testing, testing. Okay, so I think my mic is okay. Okay, so now it's already 8 o'clock. Okay, so we will start our class now. So for today, we already end chapter 2 last week, right? So now actually should be last week I say I will start chapter 5, right? But for now I already complete chapter 3, so now I proceed to chapter 3. Okay, so chapter 3 is the plasma membrane. So it is very short chapter. So I think I can finish up chapter 3 by today using these 2 hours. So actually this is very short chapters, and also you already learn in SPM also lah, you already learn also. But just there are some extra information that you not learn in SPM. Okay, so without further ado, so there are main two topics lah. About first topic is about the characteristic, structure, and function of the plasma membrane, and then the second topic is about the transport of substance across the plasma membrane, right? So so first of all, we will learn about the structure of plasma membrane. So structure of plasma membrane. Okay, so who proposed the model fluid-Mozart model? We know that our plasma membrane is categorized by fluid-Mozart model because it looked like fluid and Mozart. So who proposed this model? Who discovered, not discover, proposed who found this model? These two scientists Singer and also Nicholson. Singer and Nicholson, these two scientists, they proposed this plasma membrane model, fluid Mozart model. I think it's 1970 like that lah, 1970 like that. So these two scientists you need to know lah who are they, because maybe in exam they maybe use these two names, so you need to know lah these two names, who are they. So they propose the fluid Mozart model. So actually you can see that this is just the general diagram for plasma membrane lah. So I think you guys are very familiar with it, right? This one this one. So actually in plasma membrane, as you guys all of you know that the main component of the plasma membrane are uh, phospholipid bilayer lah. Phospholipid bilayer. So there are two layers, right? The phospholipid are arranged in double layer. So what are the phospholipid? Actually, you already learned in chapter 1, lipid part. So the structure of phospholipid. So I don't want to talk more lah. Because you already learned in chapter 1, I already teach you. And also another main component is protein lah. There are many protein that embedded within the phospholipid. So they are either integral protein or peripheral protein. So integral protein is that embedded, that can pass across the phospholipid one. Peripheral protein is located outer layer or inner layer one. It will not embedded in within the phospholipid. And also we know that there are glycoprotein and also glycolipid. Glycolipid is the glycolipid. So glyco means that it is the carbohydrate chain lah. So if there are the carbohydrate that are attached with the lipid part of the phospholipid, so it is the glycolipid, and when we attach to the protein, we call as high glycoprotein. And what else that you can found? Another one is cholesterol. Some phospholipid also contain cholesterol. So these are the few major elements that can be found in the plasma membrane. So if you exam want you to draw the plasma membrane fluid Mozart model, you need to draw like this, but don't need to draw the 3D one. Actually draw the 2D is already okay. This one is it occur in 3D. Now we proceed to the what is mean by fluid and Mozart. Apa itu fluid? Apa itu MOSFET? Kenapa kami cakap itu phospholipid, the plasma membrane is fluid MOSFET model? Kenapa kami cakap plasma membrane itu fluid? So fluid, as we know, that is liquid, right? It look like liquid. Dia macam liquid dia. And then MOSFET I think awak boleh nampak kan, itu lantai MOSFET lantai, dia nampak macam MOSFET. So why it say like that? So normally because we say fluid because of the presence of the phospholipid. Because the phospholipid and protein molecule, some protein molecule can move around within their layer. Meaning that actually our phospholipid that at the plasma in the plasma membrane is not just original fixed at there only. Dia boleh bergerak ke mana-mana punya. But at the same layer lah, within their layer. Maksudnya di sini dia akan bergerak, maybe phospholipid ni dia akan bergerak ke sini. So dia akan bergerak punya. Dia boleh bergerak punya. So it will see like that. It is look like the fluid lah. Kerana dia boleh bergerak. Dia macam awak boleh macam ni lah. Dia boleh bergerak punya. So, okay. And then, what about MOSFET? MOSFET is the presence of the protein molecule in the plasma membrane. Because the protein molecule is scattered in the membrane. Scatter means that it is located randomly. It does not have the fixed location one. The protein maybe it can locate at here, it can be locate at here. It does not have the fixed location one. It can locate anywhere in the within the phospholipid, either within or either at the outer layer or inner layer. So outer layer normally is face to the outside one face to the outside environment one, and another inner layer is face to the cytoplasmic. Okay, so this one will give the MOSFET pattern where the protein are constantly changing as they move about like iceberg in a fluid sea of the phospholipid. So that is why that when we talk about why it look like fluid and MOSFET, so these are the reason lah, if we want to explain. So maybe in exam dia akan keluar. Okay, so now we talk about the phospholipid properties in water. So actually, if you see that, why our phospholipid in plasma membrane will exist in bilayer. Why it cannot say that only can exist in single layer, but it exists in bilayer. So as you guys know, why it exists in bilayer. So as you guys know, our phospholipid is emphatic molecule, right? It contain hydrophilic head and also hydrophobic tail region. So what is mean by hydrophilic? Hydrophilic is the part that it is like water one. It can face to the water part. And hydrophobic mean it does not like water. It against the water one. So this one I just explained. So one end of the phospholipid is associated freely with water. So this one is hydrophilic head. And another part is does not free and associate with water one. So the most stable orientation for them to assume in water result in the formation of a bilayer structure. So normally in our cells, we know that our cells contain mostly water component, right? Even our cytoplasm component and our outside cell environment normally is all together contain water, right? Contain water. So outside the cell normally is interstitial fluid or lymph. So interstitial fluid and lymph component are normally also is water. Water. So if you only exist in single layer, meaning that one part of the tail or either head is cannot be achieved because both of them both environment contain water. Outside pun ada water, inside pun ada water. So if it exists in single layer, it will become like that. Kalau exists in single layer, it will become like detergent in water one. So this one is not very stable. Tak memang tak stable punya. You imagine lah, our cell, our plasma membrane very big, but it become this shape like this. Is it stable or not? So if you want to become a stable structure, so you need to become a bilayer structure. And also, phospholipid tend to have uniform width. Their roughly cylinder shape allow them to associate with water most easily as a bilayer. So compared to the detergent in water, if it it become like that, become not in uniform width, if like that, actually it is not very stable and also cannot easily associate with water lah. Because you need to know that our outermost layer and also inner part of the cell also contain many water component. So if it arranged in spherical form, actually it is very not stable, unstable, and it is very easily to dissociate. Very easily to dissociate, and then if plasma membrane dissociate, meaning the cells also break down already, especially animal cell, because animal cell only contain plasma membrane. If a plasma membrane already break down, normally the animal cell also will die, because all of the water component will go to the inside, and also many things will be affected. So that is why that the phospholipid will be arranged in bilayer form. So these are the reasons. Two reasons. There are these two main reasons. So you need to know. Okay, and another one is plasma membrane are two-dimensional fluid. So that is what we call as fluid. So actually this one I already explained. I already explained that our phospholipid layer can be free to rotate and can move laterally within the single layer, right? This one I already said. So meaning that within the single layer, you can see that this this plasma membrane is located here at the certain time it will become to here. Dia akan tukar-tukar tempat punya. Boleh tukar-tukar tempat at the same layer, within the single layer. Misalnya dekat sini semua ni dia akan tukar-tukar tempat punya. Because why? Because phospholipid are crystal-like structure in the lipid molecule to form an order array with the heads on the outside and the fatty acid chain on the inside. So this is the explanation lah. Normally because of the properties of the hydrophobic lah. Because hydrophobic it can become crystal-like structure. Crystal-like structure because it contain hydrophobic and hydrophilic region. So that is why that it can become crystal-like structure, and also in order that array with the head and outside of the fatty chain. Okay, so this is the explanation about the fluid. Okay, and also this is the plasma when we talk about the protein. So when we talk about the protein, so do protein embedded in a biological membrane move about? Meaning that this one is to determine that want to know that is it our protein molecule can be moved around or not. And we know that phospholipid can move around, but for protein can or not. So there are one experiment that conducted by Free and Eden. So these two scientists that conducted this experiment by using the different organism cells, human cell and mouse cells. So it used the human cell plasma membrane and also mouse cells plasma membrane. And then when these both cells of the plasma membrane are fused together, they found that the protein will be randomly distributed. It will be randomly distributed. So that this means that our continuous plasma membrane that surround the joint cell. So after a short time, mouse and human protein become randomly distributed through the membrane. So what are this mean that so that although there are different organisms, but the both different cell, mouse and human cell, can intermix over the surface of the hybrid cell. So the protein of each species had become randomly distributed through the entire hybrid plasma membrane. So this demonstrated the protein in the plasma membrane can move. Maksudnya dia boleh bergerak ke? Kenapa? So if let's say our protein is cannot move, meaning that actually once it fused together, the protein location is still in the same one. It will not randomly distributed one. Kalau maksudnya kalau dia dekat kanan, maksudnya kalau human cell dekat kiri, mouse cell dekat kanan, memang our fuse kat kiri punya hanya ada human cell, kanan punya ada mouse cell. But after that, when they fuse together, they found that all of the membrane protein for the both different organism is randomly distributed at the plasma membrane. But very randomly distributed. So meaning that the fluidity of the lipid, so if what we call lipid, it means the phospholipid lah, in the membrane allow many of the protein to move, producing an ever-changing configuration. So this because of the fluidity of the phospholipid. Because of this, it allow the many protein can bergerak freely. So that is why that if you want to say that is it protein can be moved around or not, so the answer is yes, it can move freely around the plasma membrane. Why? Because of the fluidity of the phospholipid. So because of this, because of the fluidity characteristic of the plasma membrane, so it can also cause the protein can move easily within the phospholipid bilayer lah. Okay. Now let us go to the phospholipid properties also. So actually phospholipid bilayer, if you see that normally either the hydrocarbon chain can be unsaturated or saturated. So if you see that when for unsaturated, so what is saturated and saturated? So unsaturated because of the double bond, carbon double bond. So if we have the double carbon bond at the hydrocarbon chain for the phospholipid, meaning that it will can form the king structure like this one. Yang akan bengkok punya. So if we have this structure, so meaning that the phospholipid bilayer will not pack to each other. So when it not pack to each other, it will enhance the membrane fluidity. Compared to this one, so this one is the saturated one. It is very packed to each other. So if it is packed to each other, meaning dia tak boleh bergerak lah. Memang tak boleh bergerak. Dia akan memang pack to each other macam orang banyak sangat lah. Orang banyak sangat. Kalau dan mereka tak ada ruang untuk bergerak lagi. So this is what we call as the viscosity by plasma membrane. So this one plasma membrane cannot move easily compared to this one. And also normally our plasma membrane also contain cholesterol. So cholesterol will act as fluidity buffer. So what does it mean? Later I will explain in more detail. So this is just I want to show you the structure lah. So there are two main structure lah. Either it can be unsaturated or saturated. So this one exam you need to mention. Not only all are saturated, some of them maybe can be unsaturated. Then they are their function lah. And also membrane protein, another structure. So I already mentioned that membrane protein can be either integral protein or peripheral protein, right? So integral protein normally are firmly bound to the membrane. So like this lah. Memang dia akan bound embedded into the phospholipid bilayer one. So these are all the purple color punya, all purple color one are the integral protein. The red one, that one is the peripheral protein, which are not embedded in the lipid bilayer. Normally are located either inner surface or outer surface. So you see that this one is the red color one. So this red color one is the our peripheral protein lah. So it really located at the outer surface or inner surface one. Okay, so now we go to the function of the plasma membrane. Okay, so we know that there are many components in plasma membrane, right? So first of all, we go to the phospholipid. So actually, if you want to refer phospholipid function, you will refer to this based on their structure. So this one I already mentioned, right? Either it's unsaturated or saturated, and some phospholipid contain cholesterol. So these are the explanation. So normally I already say that in unsaturated carbon, if the hydrocarbon chain is unsaturated, so it will form the kin structure, right? So this one it can prevent the packing of the phospholipid together. So meaning that there are many surface lah. It has the free surface so that the plasma membrane can be enhance the membrane fluidity lah. So meaning that the phospholipid bilayer can be freely move around within the single layer lah compared to this one viscosity plasma membrane. This one is only contain saturated one because no double bond at all, only single bond. So normally it is very packed to each other lah. Dia macam awak boleh cakap dia macam ice lah. Kalau awak tengok solid punya atau ice punya, dia memang pack to each other so they cannot move lah. Dia tak boleh bergerak kan, tak ada ruang untuk bergerak. So this is what we call as viscose plasma membrane. So normally viscose plasma membrane because it cannot move. So the fluidity of this type of plasma membrane is very low lah compared to this one. Okay, another one is cholesterol within the animal cell membrane. So normally animal cell contain the cholesterol within the phospholipid bilayer. But some of the cell, plant cell, fungi cell is rarely have, normally only have in animal cell. So normally cholesterol, the function is to reduce the membrane fluidity at the moderate temperature. So normally it acts as the fluidity buffer. Fluidity buffer means that it can based on the temperature to regulate the fluidity of the membrane. So if in the moderate temperature, it will reduce the membrane fluidity. Because if the fluidity of the membrane cannot be too high or cannot be too low. If it is too high, meaning that it can easily to be burst. Right? Kalau dia bergerak terlalu bergerak terlalu rapid, move too rapid and also too soft compared to weakest one. Okay, but at the lower temperature, it will hinder the solidification by disrupting the regular packing of the phospholipid. So in very low temperature, so if you guys know that in very low temperature, meaning that they will become packed to each other, right? Like solid, right? So if solid particle, you lower the temperature, the solid particle is very packaging to each other, very tightly to each other. So this cholesterol function is to prevent the solidification, prevent the phospholipid bilayer to pack tightly to each other. It can maintain the fluidity of the plasma membrane by disrupting the regular packing of the phospholipid. So it will prevent the packing lah. Tak mau pack to each other. Dia akan ada lubang untuk maintain the fluidity. Okay, so these are all the explanations. So actually you can pick yourself. Okay. Okay, now we go to the protein function. Okay, so we know that our plasma membrane contain integral protein and also peripheral protein, right? So normally the protein will be function integral protein lah. Integral protein will mostly take part in the function, right? So actually there are many functions in protein. So if you see that one of the first function is transport protein. I think everyone know, right? Passive transport and also active transport. So this one later I will explain it. So I don't want to focus here. So we know that there are two main types of transport, right? Active transport and passive transport. Okay. Another one is enzymatic activity. So as we know that protein can also become enzyme, right? So maybe some of the protein can become enzyme. They are jadi enzyme. So it will take part in the enzymatic activity lah. So the substrate molecule will bind to this enzyme and then to release the product lah, hydrolyze, have the enzymatic reaction in this protein. So this protein is not for the transportation, but for the enzymatic reaction. Another one is signal transduction. Okay, so what is signal transduction? You can see, you can see here. So normally this one is that the membrane protein is act as the receptor. Our membrane protein is act as the receptor to receive the molecule, to receive the specific molecule that can fix the chemical messenger. So normally this one is normally found in the hormone lah. So if you guys know that for your senior, sem 1, sem 3 senior, if you got you know that in your sem 2, you learn about mechanism in steroid hormone and mechanism in non-steroid hormone, right? Especially in non-steroid hormone, right? And non-steroid hormone, they are one of the receptor, right? Only the certain receptor like insulin and glucagon, non-steroid hormone one, non-steroid hormone one, it will bind to this receptor, right? I don't know you guys remember or not. If you don't remember, you have to take the U2, you should know it. But for your junior, you will learn in semester 2. But I just say that I just want to give the message that for this one, it will act the protein will act as the receptor and will receive the signal molecule. So for example, like hormone lah. So I give the example like insulin. So insulin. So when the insulin is bind to this receptor, so this receptor will send the signal to the cells. So this will may cause the protein to change the shape, allowing it to relay the message to help the inside of the cell. So this one will send the signal lah. They akan send the signal by changing the protein shape. So usually by binding to a cytoplasmic protein. So it will send to the signal to the protein within the cell, and then will have the cerebral reaction within the cell lah, to like insulin, or like insulin it will have the reaction to change the reduce the blood glucose level, right? So it will reduce the blood glucose level, reduce the intake of the glucose molecule from the cell. So this one is the signal transduction. Why we already say like transduction. So it give the signal to the cell. Once it receives the signaling molecule, bagi signal lah. Okay. Another one is intercellular joining. So this one is that the membrane protein of the adjacent cell may hook together in various kind of junctions, such as gate junction and tight junction. So this type of binding is more lasting than shown in the so this one actually is the binding signal molecule binding actually is very short. Once the situation is already okay, this signal will be released out. Once it does not have any signal molecule, then the binding will be not binding lah. The receptor will not bind this signal and then to remain normal lah. But for this one will bind to the cells and cells for very long lah. So this one joining, normally you can imagine that the animal cells are joining together. So normally this one are the example. I don't know you guys got see or not. So it got say that tight junction and gap junction, right? So you see here, this one is about the example of the microvilli in the small intestine. So normally tight junction is the, if you see that tight junction is like this one. Memang dia sangat, kenapa dia cakap tight? Memang dia sangat tight lah. Dia akan the plasma membrane and plasma membrane are joined tightly one. So this plasma membrane of the neighboring cell are very tightly pressed against each other, bound by the specific protein. So this protein lah, ini protein dia lah, bound by this specific protein. And then forming the continuous seals around the cell, tight junction establish a barrier that prevent leakage of the extracellular fluid across a layer of epithelial cell. So normally this one, how this one is to prevent the extracellular fluid flow away from the cell. So normally this one is very tight junction. So why we call as tight junction? Because it is very tight to each other. Dia macam ikat lah. Ikat punya. Dia ikat sekali punya. The plasma membrane dia ikat sekali. Untuk takkan sekali lah. Jangan dia lari tempat. And then another one is gate junction. So you see that gate junction gate junction, see this structure. So this structure is not very tight to each other lah. So you see here, memang dia akan, memang dia punya ada banyak, ada spacing lah compared to this. So gate junction are also called communicating junction. This one is provide cytoplasmic channel from one cell to adjacent cell. So normally this one is to exchange material lah for between the cells. I want to exchange the material between the cells one. And then in their function to plasmodesmata. So it has the same function like plasmodesmata to exchange the material between the cells. Okay, so that's just like that. So this I just want to you what is mean by tight junction and gap junction lah. So this one junction, how to it made because of this protein, because the protein within the plasma membrane. If no this, memang junction tak ada akan berlaku lah. Okay, another one is act as the anchoring function. Anchoring function is that it will attach to the cytoskeleton and also extracellular matrix. So what are this main purpose is to provide the mechanical support to the cells. So microfilament is one type of the cytoskeleton, right? So cytoskeleton we have microtubule, microfilament, and also intermediate filament, right? So I hope that you guys still remember what we learned in chapter 2. So this function is to maintain the cell shape and also stabilize the location at certain membrane protein. So as we know that the cytoskeleton function is to provide mechanical support to the cell, right? So how it provide? Normally it can attach to the plasma membrane. So it can to maintain the cell shape because our plasma membrane normally especially in animal cell, it provide the cell shape to the animal cell, right? And also to stabilize the location of the membrane protein. Make the membrane protein cannot move around. Although membrane protein can move around, but it cannot tak boleh lah sesuka hati nak gerak ke mana-mana kan. So actually it need to has also the some fixed shape lah, fixed position. Bukan sentiasa akan bergerak sini, bergerak sana. Kalau dia sentiasa bergerak sini, bergerak sana, macam mana dia nak function ni. Ni macam mana function. Especially intercellular joining. Because intercellular joining, the protein cannot be moved around. So this one normally is to stabilize. So it uses stabilize to stable the location of the membrane protein. And then the protein that can bind to the ECM molecule can coordinate extracellular and intracellular change. So this one, actually this is the full image lah. So actually we know that it say the ECM, right? So what are the ECM? So ECM normally is component of these three components lah: petroglycan, collagen, and also fibronectin. So normally these are all the main component in ECM. So fibronectin, this one actually is to provide the anchor lah to the ECM one. Dia satu jambatan lah. You can say that dia macam it is look at as the bridge from the plasma membrane to the ECM. So it join the between the plasma membrane and ECM. And also collagen fiber are embedded in the bed of petroglycan complex. And then petroglycan complex consists of 100 of petroglycan addressed non-covalent to a single long polysaccharide molecule. And also we have intergreen. So this one actually is about the what that we call about the ECM, extracellular matrix. So normally this one is work with the cytoskeleton to provide the work with the cytoskeleton, especially microfilament. Provide the maintain the shape of the plasma of the animal cells and also stabilize the plasma membrane. So cytoskeleton normally is within the cell, support within the cell, and extracellular is outside the cell one. So normally you will see this structure lah. Normally, petroglycan complex is looked like this. Okay, so this one is about the ECM. So If you want to ask me want to know or not, actually you should know. Kena tahu lah. Mana tahu dia cakap explain the protein function. So you need to not just say like this. Some main important you should say lah, but for this is the in detail one lah. Okay. And another function is to cell recognition. For this one, actually, is very important in immune response. So for your senior, you should know already cell recognition. You already learn semester 2 immunity chapter. Yang ni pun ada berkait dengan semester 2 punya. So I just give this example lah. So normally our antibody our antibody is also the one of the glycoprotein. So cell recognition is involve the glycoprotein punya structure. It is not the protein bukan protein saja. They are involved glycoprotein, meaning that they are attached of the carbohydrate at there. So this normally these are the example. So this is the example that later you will learn for junior semester 2 awak akan belajar semua ni. But you don't need to know it now. Ini yang senior awak kena tahu lah. Dan junior awak tak perlu tahu. You will know in your future. So this one is about the immunity application lah. Application of this cell recognition. So normally this is about the immunity. So you see that awak tengok macam ni kan. Ini plasma membrane, right? So actually in our plasma membrane it will have the what we call as the antibody lah, an antigen receptor. This is what we call as antibody structure lah. Ini antibody structure. So our antibody actually is the glycoprotein one. It is one type of glycoprotein, and it will involve in cell recognition. So cell recognition meaning that there are some of the antigen for immunity. There will antigen will bind to this antibody one. Dia akan bind punya. Okay, and also for this one is helper T cell, helper MHC molecule. So this one MHC molecule also act as the glycoprotein. And then for this one is about the T cell antigen receptor. So this one is also one type of glycoprotein. So for senior, you should know already what are the mechanism. Kena tahu lah. Kalau if you especially for those who retake, you do. But for those who don't retake, okay, you can forget it because you don't retake. Okay, but for those who retake, you should know lah all this. Okay, I just want to give the example lah, cell recognition punya. So normally it is act as the identification tag that are specifically recognized by the membrane protein. So it is act as the identification tag. Dia macam bagi signal punya. Kalau dia ada kalau ini macam ni, hanya certain antigen boleh bind dekat sini sahaja. Kalau ini macam IgE antibody, maybe only the pathogen that have the antigen that fits with this antibody receptor, antigen receptor can bind to it. If not, cannot. And this one also same. So what is called as cell recognition? So there are two different cells. Ini satu cell. And then this cell is important to play a bagi signal punya. They are not bagi signal, identification tag. It will act as the identification tag to make the this T cell to identify this cell. Dia macam kenal lah, nak kenal awak. Senang cerita lah, kenal awak. Kerana T cell ni dia memang tak kenal siapa pun. Dia tak kenal siapa pun. Tapi kalau saya nak kenal awak, memang saya akan bind dekat awak lah. Saya akan bind dekat awak. Okay, apa awak ni? Because this one is play important in immune system, but so this one is bind to the antigen. So this MHC molecule so that it will know this type of antigen have what apa shape dia macam mana, lepas tu dia apa property, karakteristik, T cell itu tahu. Tahu lepas tu dia akan play the role in the mechanism of the immunity lah. So it will already know this pathogen. So once it look that, or dia keluar buat rentaan di our blood. Lepas tu dia tengok ada sama punya, yang ni cakap, yang ni saya tahu. Ini orang ni macam itu penjenayah lah. Saya tahu saya akan tangkap awak. Saya nak bunuh awak. Nak tangkap awak. Macam polis lah. You can imagine it is look like a police. Polis dia kena tahu. Oh, ini penjenayah A. Okay, saya dah tahu lah. Lepas tu awak keluar. Buat bendaan sesuka hati. Lepas tu awak tengok. Suddenly awak tengok penjenayah A. So awak try to tangkap dia kerana awak tahu. So that is why we say as cell recognition. It will provide the identification tag, identify. It make the cell to identify him. So normally this one is very important in immune response lah. So this is just the basic lah. So for junior, you don't need to know this all. I just want to explain for you to understanding lah. Make you understanding what is about the cell recognition. And then this one I just sharing. Kerana yang ni sepatutnya chapter 2 kena belajar lah. You already learn in chapter 2 and also I already explained in chapter 2. What are the between function protein synthesis, how the protein can be synthesized. So this one I don't want to explain in detail because I already explained in semester 2. So that's all. This one I just want to highlight some part about the glycoprotein one. So normally, if you see here, some certain protein, normally it will be secreted out through the exocytosis process. Some of them will bind to the surface of the vesicle. You will see here. So secretory protein, membrane protein, and lipids are synthesized in the ER. In ER, carbohydrate are added to transmembrane protein, making them glycoprotein. So actually, our glycoprotein in plasma membrane actually already formed in the transport vesicle. Already formed in the transport vesicle by ER. So in the ER, our carbohydrate will be added to the transmembrane protein already. So our membrane protein structure actually already formed it will form very awal dekat transport vesicle already inside the ER, endoplasmic reticulum. So after that, it will go to the Golgi apparatus for the modification, and then it will form the secretory vesicle, and then after that, some of the protein that within the vesicle were secreted out, and then the membrane protein that already bind within the vesicle will become the part of the plasma membrane protein lah. So that is what we call how the glycoprotein and glycolipid protein and also the plasma membrane protein form. So I just want to highlight this lah. Others I already teach, so I don't want to wasting the time at here now. Okay. Now I will proceed to the transport in plasma membrane. So about the structure of the plasma membrane is done now. So in examination, if you want to explain about the function and the structure of the plasma membrane fluid Mosaic model, all of this you need to explain lah. Semua ni memang awak kena explain. Kerana ada satu tahun dia keluar 15 markah kan. If you see that one partial question, it come out one question 15 marks, and also want you to draw it. Want you to draw and then explain the structure and function of all of this. So kalau nak explain memang semua ni kena explain bah. Okay, so that's all for structure and function. Next, I will go to the transport. it. So any question or no? Ada soalan tak? No question. Okay, so now I will go to the movement of substance across the membrane. Okay, so for transport in plasma membrane, okay, so normally if you see here, when we talk about the transport of plasma membrane, we will have the two main type of transport, passive transport and active transport. So passive transport normally is with both simple diffusion, facilitated diffusion and osmosis. Active transport yang paling famous, sodium potassium pump. And then we also have to learn bulk transport. So maybe this one is new to you, but although it's new, actually you already learned about endocytosis and exocytosis. And then we have phagocytosis, spinocytosis. And we have another one is about cell-mediated endocytosis. So we will learn this one by one. So now we go to the simple diffusion. So actually simple diffusion is very simple because we already say that it is very simple, right? So normally this one is only involve the phospholipid bilayer. The molecule that can be diffused through the phospholipid bilayer normally we call as simple diffusion. So simple, this is the simple diffusion definition. So normally it is the movement of small molecule from a high concentration region to a low concentration region, down concentration region. So normally these all of the passive transport all are down concentration gradient. So simple diffusion is that the molecule molecule normally is involved can be passed through the the phospholipid bilayer lah. So normally these molecules are very small and uncharged molecule. So normally like oxygen, carbon dioxide, water molecule, and also small uncharged molecule can be passed through it. So some big molecule like glucose, lactose, protein, amino acid, those are there, they cannot pass through using the simple diffusion because it is very large molecule. So okay, so this is the simple diffusion, very easy. Another one, facilitated diffusion. So when we see about facilitate, so what is mean by facilitate? Cepatkan, kami nak cepatkan diffusion dia. So meaning that this facilitated diffusion will involve the protein. It will involve the protein one. So the molecule that cannot pass through the membrane, they cannot pass through the membrane, they will involve the protein. Now that is what we call as facilitated diffusion. So thus the difference between the simple diffusion is that it will involve the membrane transport by the protein one. The transport is by the membrane protein, and also it is down concentration gradient from high to low one. So normally there are two main types of protein that can be involved in facilitated diffusion, while either one is channel protein or carrier protein. So channel protein, some of the book say core protein, so core protein and channel protein are acceptable. So normally the molecule are large molecule, polar molecule, ion molecule. All of them can be passed through. So you see here, there are sodium and potassium, but you say that sodium potassium is active transport. But sodium and potassium not necessarily will involve in the active transport only. It can be also involved in facilitated diffusion. So for those who are you are senior, more senior. So if you know that actually semua memang basic punya lah. Dan memang basic kerana semua topik akan relate kepada semester 2 or semester 3 that you learn one. Yang ni you will learn in semester 2 also about the nerve transport, nerve impulse, transport of nerve impulse. So actually, you will learn in semester 2. So you will know lah what I mean by why the sodium and potassium can be involved in the facilitated diffusion. So not so necessary that sodium and potassium can only involved in active transport. It can also be involved with the facilitated diffusion. So all the molecule lah. Boleh cakap semua molekul lah. Normally large molecule, ion molecule, glucose, amino acid. Normally all of these can be transport using this one. Using this protein. channel protein or carrier protein. Okay, so normally channel protein and core protein, what are the difference is channel protein is it contain the corridor lah. Dia tengah-tengah dia ada satu jalan punya lah. Memang ada satu jalan punya. But for the carrier protein dia bukan, it will change the shape one. It will transport based on the shape. So carrier protein you need to know not necessarily only involved in active transport. Carrier protein also can be involved in facilitated diffusion one. Just either want to see lah, either is down concentration gradient or against concentration gradient. If it is down concentration gradient, so no, it is confirmed facilitated. But if it involves against concentration gradient, it will involve active transport. So in objective question, so don't want to say that if you statement say that carrier protein only involve active transport, this statement is wrong, false. Some of them say true. Most of you I tahu. Because semua cakap, oh carrier protein active transport. Lepas tu carrier protein tanya channel protein sahaja involve facilitated diffusion. So no, both protein can be involved in facilitated diffusion. So what are the difference between these two? Normally carrier protein can change the shape. It move the molecule by changing the shape. Kalau satu molekul bind dekat sini, dia akan tukar shape dia dan release the molecule lah. But for this one, dia tak ada change shape dia, memang dia akan satu jalan yang free punya, akan masuk je. Dia akan masuk je. Okay, so this one is about the facilitated diffusion. Okay, now it's about active transport. So this one is most important lah. Because active transport is very special compared to the simple diffusion. Because simple diffusion is down concentration gradient. If it involve down concentration gradient, it will not involve energy. Dia tak perlu energy punya. Kerana dia dari banyak ke kurang kan. So if banyak ke kurang, tak perlu energy. Memang tak perlu energy, dia automatically akan boleh berlaku. But for active transport against the concentration gradient, meaning that the molecule or ion will move from low concentration to high concentration region. So when it involve this, so memang dia kena ada power punya. Dia memang kena ada satu power. Kerana dia tak boleh automatically open punya. Memang dia akan kena satu power, so it involve metabolic energy. Dia memang kena ada satu energy. So yang paling famous sodium potassium pump lah. So ini salah satu example lah. Okay, now we go to okay, so active transport, it will involve two types of transport. Either it is antiporter or symporter, and another one is uniporter. I don't say letak sini. Okay, antiporter, symporter, what are they? So if you see that antiporter is that there are two molecules that go pass through the membrane but in opposite direction. Maksudnya maybe A dia masuk carrier protein and then keluar dekat B. But at the same time, the B molecule from the inner cell, it will also pass through the carrier protein to the outer cell. So there are two different molecule of ion can be passed through the membrane but in different position, different direction. Satu masuk, satu keluar. Satu masuk, satu keluar. Like sodium potassium pump. Sodium potassium pump is the one example of the antiporter. Ada keluar, ada masuk. And then another one is symporter. So symporter is that it also involve two molecule but dua-dua masuk. Either dua-dua masuk, either dua-dua keluar. In both are in the same direction one. Dua-dua masuk atau dua-dua keluar. And another one is uniporter. Uniporter is that only one particle only. Hanya satu particle je. Either dia masuk atau keluar. But it only involves satu particle. Symporter involve dua particle. But in the same direction. Antiporter involve two particle but in different opposite direction. Satu keluar, satu masuk. Yang ni dua-dua keluar atau kedua-dua keluar masuk. Uniporter satu keluar atau satu masuk. And both these three of them require energy because it active transport. Memang active transport dia kena ada energy. So these are the three ones. So antiporter, one of the example, most famous sodium potassium pump. And for simpleter, later you will learn it. Yang ni punya. Hydrogen ion and sucrose, hydrogen ion and sucrose pump, or also sodium ion and glucose pump. Yang ni lah example of the simpleter. So actually, I think this one is new, and some of you not focus, but you need to know. Memang kena tahu what are they: antiporter, simporter, and uniporter. Okay, and then now we go to the sodium potassium pump. Okay, I think this one you already learn in STPM BIO. Memang kena learn. So I just explain, or also, but for now I explain. Okay, maybe you already forget, so I explain in detail. So please hear carefully. So normally, if you know that sodium potassium pump, so as you guys know, normally awak kena tahu punya satu, memang dia akan ada ATP. And yang paling penting, yang special punya adalah potassium ion will be three potassium will pump out and two potassium will pump in, right? Kalau awak tahu kan, three potassium ion from inside the cell will pump out to the cell, outside the cell, and two potassium ion will pump into the cell. Ini semua tahu kan, basic punya. Three sodium keluar, two potassium masuk. So yang ni awak tengok, so these are all the mechanism. So firstly, our sodium ion, so when it achieves the condition like this, so I already say that octane transport is against concentration gradient. So the sodium ion inside the cells must be very low lah, and outside the cell must be very high lah. Right? Okay. So when it achieves in this kind of condition, the sodium ion will start to bind to the active site of the carrier protein. Cytoplasmic sodium ion will bind to the sodium potassium this carrier protein with high affinity when the protein has this shape. Dia akan tukar shape macam ni. Lepas tu sodium ion will bind to it. Okay, so all together will contain 3 sodium potassium ion will be bind to here. After that, it will stimulate phosphorylation process. So what are the phosphorylation process? Apa tu? It will stimulate phosphorylation process using ATP molecule. So dia menggunakan ATP lah. So when it require ATP, meaning that phosphate group will bind to the carrier protein. So what we call as phosphorylation. What are the definition of phosphorylation that you learn in chapter 5? So phosphorylation meaning that it have adding one phosphate group lah. The protein, this protein is binded with the phosphate group from ADP punya. This phosphate group from ATP, so that is why it requires energy. So when it bind already, so this phosphorylation will cause the protein, carrier protein, change the shape. So when it changes the shape, it will reduce the affinity of the sodium ion. Kerana dia sudah tukar shape dia, it will already change the shape. So this carrier protein is actually not affinity to the sodium ion already. So the sodium ion will be released out from the carrier protein and released out from to outside the cell. Okay. Now when it changes the shape, now it is affinity to the potassium ion. Just sekarang suka potassium ion lah. It already have the same shape with the potassium ion. So two potassium ion will bind to the this protein, carrier protein, bind to this protein. And then when once the potassium ion bind to this protein, it will release the phosphate group. Dia akan release the phosphate group. So once the phosphate group release, it will change the shape like this. Change to the shape in with the change to the origin shape lah. Macam ni punya. Macam ni punya. So when it change the shape, so similar from the sodium ion, the binding site already not affinity to the potassium ion already. So once it is not affinity to potassium ion will release from the protein and release into the cell lah, cytoplasmic cell. And then so two potassium ion is released, and then it will repeat the process again and again. And then 3 potassium ion will bind to it. Then the ATP phosphorylation, phosphate group bind to the protein, and then they are going to cut shape and then release sodium ion at the same time. After one it release, dua potassium ion akan bind, and then it will trigger the release of phosphate group, and then they akan change the shape again, and then it will release the potassium ion, and then it will repeat and repeat and repeat. It will repeat the process unless the condition is achieved this kind of condition again can work against concentration gradient punya condition. Okay, so that is all about the sodium potassium pump. Okay, now we go to the core transport. Okay, core transport, what are meaning by core transport? So core transport normally is involve the energy not from ATP one, bukan dari ATP. It is indirect active transport. Faham tak? Yang ni dia menggunakan ATP, directly guna ATP. But for this, it does not involve ATP. It will involve indirect active transport. So normally the energy is come from the electrochemical gradient. Electrochemical gradient. So what are these electrochemical gradient? So normally, if you want to know that what is electrochemical gradient, actually this one is under semester 2 syllabus lah. Vesting potential. Senior sudah tahu kan? Vesting potential. Action potential, vesting potential in semester 2 lah. So memang dia agak berkait punya. Memang semua berkait punya. So itulah. So if I want to explain, can be explained maybe panjang cerita punya. So I don't want, because I don't want to be you guys confused. So normally if you want to about the what is meaning by core transport, so normally the energy from the core transport is come from the electrochemical gradient ini. Electrochemical gradient. What does this mean? So what does the electrochemical gradient mean? Normally if you see that our cells yang ni, cells ni, memang our electrical balance and also concentration balance are not constant. So that is why that you see that why 3 potassium pump out, 2 potassium pump in. Kenapa bukan 3-3 atau 2-2? Kenapa dia ada pasal perbezaan punya? This one kalau dia sama, memang sama. So we don't have the action potential and vesting potential. We don't have the action potential and vesting potential. Semua macam vesting potential sahaja. Macam vesting potential and no action potential at all. If no action potential, saya cakap cerita what is mean by action potential. Action potential meaning awak macam ada signal lah. Dia signal untuk awak. Macam awak panas punya signal, awak nampak punya signal, semua tu lah. All of the signal inside the cell. So if know that, normally you will not have any signal. Awak macam orang yang tak ada signal punya lah. Saya cakap dengan awak, saya tak boleh dengar, memang awak tak ada macam-macam punya signal lah. Your nervous system is already destroyed. So this one, so because of the different of the chemical gradient, the electrical impulse, and also concentration gradient between the cell, outside the cell and inside the cell. So it will create the electrochemical gradient. So this one electrochemical gradient will provide energy. Dia boleh provide energy punya. It will can provide energy punya. Okay, so for this kind of transport, so if you see here, so this one is hydrogen ion supercore transport. So normally, this one actually this is the full process. So normally, firstly, dia akan guna potong pan. Memang dia akan guna potong pan. So for here, it will already involve ADP. And then it will pump the hydrogen ion from the cytoplasmic part into the outer part one. Okay, now it contain many hydrogen ion at the extracellular fluid. So this one will cause the charge, particle outside the cell is highly positively charged, and then inside the cell is already negatively charged. Because all the potassium ion is already semua keluar. So this one already create the electrochemical gradient already. Dia sudah create a electrochemical gradient. And then this hydrogen ion dengan sucrose molecule, it will help this hydrogen ion will help the sucrose molecule akan pass through the this protein. So this core transport protein together, and then this one will create down the electrochemical gradient. So you see here, neutral solutes such as sugar can be loaded into the plant cell by core transport with hydrogen ion. So this one play a key important loading to sugar, pass through the hydrogen ion together one. And then you will see that hydrogen, and then the core transport mechanism involve hydrogen ion also participate in regulating. Yang ni lain cerita lah. So if you see that the sucrose molecule and also hydrogen ion will pass together. So this one is one type of what protein? What potter? Syn-potter or antipotter? Syn-potter or antipotter? So this one is what we call as syn-potter one. Yang ni. Because both are them in same direction. If you see that both antipotter and syn-potter use the potential energy of electrochemical gradient form the chemical ion, hydrogen ion or sodium ion. Why it will so this it will act as the energy, it provide the energy, so it can help the sucrose molecule to transport from the outside into the inside. Dia akan bantu plant cell ni transport the sucrose molecule from the outside cell into the inside cell, inside of the cell. So energy datang dari mana? Energy datang dari mana? Hydrogen ion. Because hydrogen ion it can diffuse down the electrochemical gradient. Yang ni. Able to use the diffusion of hydrogen ion down its electrochemical gradient into the cell, drive the uptake of the sucrose molecule. So although not technically of the core transport process, so the ATP-driven proton pump is shown here bottom, which concentration of hydrogen ion outside the cell. So the resulting hydrogen ion gradient represent potential energy that can be used for active transport for glucose. So ATP hydrolysis indirect provide the energy necessary for the core transport. So the ATP actually is not directly used, but it indirect use. Dia macam indirect lah. ATP dia tak direct macam ni. Yang ni dia direct dekat protein ni. Tapi yang ni sucrose and hydrogen ion pump ni dia tak direct punya. Dia guna proton pump, lepas tu ada hydrogen ion, it create a electrochemical gradient, and then it will give the potential energy to the sucrose molecule so that the sucrose molecule can use this energy to pump into the cell. So same as this. This one is about the sodium ion and glucose molecule. So once the sodium ion bind to the carrier protein, so it already provide the electrochemical gradient. It will already provide the electrochemical gradient. So it will create the potential energy to the glucose so that glucose molecule can be bind to the carrier protein and then transport into the cells. So normally for this one, the ion that involved in the potential energy that already create the electrochemical gradient normally is down concentration gradient. Down concentration gradient. Dia akan pergi high concentration region to low concentration region. Hanya sucrose molecule or glucose molecule akan bergerak from low concentration to high concentration gradient punya. Ini awak kena tahu lah. Dan tak sama macam ni. For ni, active transport yang ni, memang dua-dua partikel akan involve from low concentration to high concentration punya. But for this, only the solute particle, either is sucrose or glucose or other amino acid, involve the low concentration to high concentration only. But the ion that act as the potential energy, provide the energy to sucrose, so ni dia akan down the concentration gradient punya, or we say the down the electrochemical gradient. Electrochemical gradient. So yang ni awak akan belajar pula dekat chapter 5, electrotransport chain, especially oxidative phosphorylation, ETC and also chemiosmosis. So yang ni awak memang kena tahu lah. Memang kena tahu. Kalau awak tak faham yang ni, memang nanti awak datang chapter 5, if you learn about oxidative phosphorylation and chemiosmosis, awak pun tak tahu dia cakap apa lah. So ini memang sangat basic lah. So apa yang saya nak tekankan bahawa energy yang dia provide is not ATP but hydrogen ion. And hydrogen ion, because it already create the electrochemical gradient, and it can provide the potential energy to the solute particle so that this solute particle can be moved from low concentration gradient to the high concentration gradient region. Sama dengan ini pun. So this is what we call as core transport. This one we also will be applied in oxidative phosphorylation and also chemiosmosis. So please, so memang kena faham lah. Memang kena faham. Ini memang basic. Sangat basic punya. So if you don't understand this, chapter 5 memang awak pun tak faham. So ada soalan tak? Siapa yang tak faham, please ask now. The protein in electrotransport chain are example of, yeah, you can say that, yeah. It's one of the example lah, core transport. Because electrotransport chain, if you already learn, although I not teach yet, you already learn, dia tak guna energy kan? Memang tak ada guna ATP, kerana dia nak hasilkan ATP. Takkanlah dia nak guna ATP lagi. Kan? Electrotransport chain is the responsibility to provide ATP, to generate ATP. Kalau dia nak guna ATP lagi, not make sense lah. So but it is one type of active transport. So if you know that electrotransport chain, the chemiosmosis is one type of active transport. So what if you one type of active transport, I already said it require energy. So kalau kamu guna ATP, mana energy awak datang? So your energy hanya boleh datang dari sini lah, electrochemical gradient. So this term you will see again in chapter 5. So if you see the Campbell buku Campbell atau buku Solomon semua tu, dia akan guna electrochemical gradient as the potential energy. So for those who still not understand electrochemical gradient, awak tak tahu lah. I already explained banyak kali lah. So what is electrochemical gradient? It's actually combination of two different properties. One of the property is about the concentration of the ion. Concentration of the ion. Karena different concentration ion. Satu low, satu high. Memang tak balance punya. memang tak balance punya. Our plasma membrane inside cell ion and outside ion memang tak balance punya. Dia tak akan balance punya. Memang ada satu low, ada satu high. And then another one, because of this, our electrical charge also different. One will be negatively charged and one will be positively charged. Akan berubah punya. Either inside negative atau outside negative. Memang dia akan ubah punya. Memang akan ubah punya. So this one also will be applied in semester 2 in nerve transmission impulse. So semua memang berkait. So ini chapter memang sangat penting. Dia bukan hanya berkait dengan semester 2, dia juga berkait dengan awak cellular respiration and also photosynthesis. Photosynthesis pun ada apply konsep ni. So that's why saya tekankan lah. Saya memang tekankan topik ni. Kerana topik ni banyak saya rasa sekolah awak tak fokus punya. I think lah. Normally they focus ni je. So dia boleh sebab core transport, I don't know your school teacher got to focus or not. But so that's why, because I know that many school teacher not focus, so I focus this. Okay, so hopefully you guys already understand. So because in chapter 5 I will not takkan explain panjang cerita macam ni lah. I will just directly explain je what is based on electrochemical gradient. So at that I will not explain one by one like this already, because in chapter 5 dia memang sangat complex. So saya tak dapat explain like this. So this is the basic. So please know. So ada soalan lagi? So I already answer your question right, Dragon Ball. Yeah. In electron transport chain is one example of the core transport. Yeah. Memang satu example. One of the example. Okay, so that is what core transport. So yang ni pun sama. Yang ni adalah yang plant punya sucrose and hydrogen ion. Yang ni pun sama juga. Yang ni pun dia guna nitrate ion and hydrogen ion to balance. Yang ni dia guna potassium ion. So yang ni memang core transport lah. So yang ni awak tengok, yang ni potassium ion, awak tengok dia memang pun tak ada ATP punya. Dia hanya solid particle, yang ni unipolar punya. Dia memang tak ada energy langsung. So energy dia datang mana? So memang energy dia datang daripada electrochemical gradient. Memang dia datang daripada electrochemical gradient punya. So, okay. So, please. Awak kena tahu. So not necessary. So in active transport punya definition, please don't write ATP. Don't say that your energy is ATP. It is not necessary only ATP. You need to use metabolic energy such as ADP. For example, ATP. If you directly use, or it will use ADP. Sorry, wrong. Definition, your definition is directly wrong. Why? Because not necessarily the energy must be ATP. It can be hydrogen ion or sodium ion, any ion, potassium ion pun boleh. Because of electrochemical gradient. So, AD transport definition, please also alert. Jangan tulis ATP only. Awak tulis ATP memang salah lah. Kerana konsep tak betul, memang salah. Siapa cakap? Memang ATP satu, tapi bukan semua. Core transport tak guna ATP. So lah, inilah difference dia. So, please differentiate between them. So, there are active direct transport and also indirect transport. Direct transport yang famous of sodium potassium pump. Core transport is indirect transport. Okay, yang ni clear? Kerana why I tekankan ni, kerana many reason lah. One of the reason is your school teacher not teach, and another reason is this is the basic concept for you when you enter chapter 5 and also semester 2, nerve impulse. So you need to follow this concept. So if you don't know, memang susah lah. Susah untuk understand lah you chapter 5. Okay, another one. Now we proceed to bulk transport now. Okay, bulk transport. Okay, so this one actually I think you guys also know, right? endocytosis and exocytosis. So bulk transport is the spatial transport where normally it involves the large macromolecule, normally like protein, polysaccharide lah. Yang memang huge punya molekul lah. Dia bukan basic monomer like amino acid, glucose ke, ion ke. Yang memang involve the huge molecule like protein, polysaccharide, and polypeptide ke, polypeptide, and also like lipid, like that. So normally it will involve into and out of the cell lah. So endocytosis is that dia masuk dalam, masuk dalam punya. Dia masuk dalam into the cell is endocytosis. Outside the cell, release the substance, release the molecule. That is exocytosis. So please differentiate them. Endo masuk, exo keluar. Exo kan, exo acid. You can say the acid lah, ex acid. So exo lah. Ex kalau ada X kan, so you can imagine that it's acid. So dia keluar lah. Okay. Okay, so so exocytosis. So one of the example exocytosis is that lah, inilah. Yang saya bagi example ini punya. Mana? Oh, bukan sini lah. Yang ni lah. Protein synthesis 1. Protein synthesis 1. If you go to the secretory vesicle, after that, after secretory vesicle fits to the plasma membrane, and then it will release the particle, right? You will start to the release particle. The two membrane will fuse to the plasma membrane, and then start to release and secrete the protein outside the cell. So this one we call as exocytosis. It already released. So normally it will involve the secretory vesicle. So you see here sama. So the secretory vesicle will start to attach to the go to the plasma membrane, and then it will fuse with the plasma membrane, and then the vesicle will become the component of the plasma membrane, and then the protein within the vesicle will be released out from the cell. So this is just the process of the exocytosis. So normally it will involve the secretory vesicle lah, yang paling penting. So the secretory vesicle is come from the Golgi apparatus. So faham kan? Okay. Okay. And then for endocytosis is more complicated because under endocytosis we have 3 different type of endocytosis here. Phagocytosis, pinocytosis, and also cell-mediated phagocytosis. So phagocytosis and pinocytosis, what are their difference? You can differentiate between one is cellular eating and another one is drink. Satu makan, satu minum. Satu adalah makan punya. Satu adalah minum punya. What does this mean? Meaning that, eating meaning that normally is involved the solid particle lah. Memang solid particle punya. Large solid particle. Pino meaning that it will involve only dissolved material. Normally it already is a very small particle one. Like the very small particle one. But for this one, it will involve large particle. Normally, it's the food or bacteria. And then cell-mediated is more spatial. It will involve the specific molecule combined with the receptor protein in the plasma membrane. So this one later I will explain. Okay, so this one is a process of the phagocytosis. Right. This one I think very famous lah. Ini sangat famous punya. Famous punya process. I think this one you already also learned in STPM by phagocytosis. It also play the role in immune response, right? One of the is immune response, and also ammoebia and fuso and paramecium also have phagocytosis, right? Because they akan makan. So normally this one is about the cells, white blood cells. So normally it will when got the pathogen atau food molecule, either food molecule or pathogen, it will surround the pathogen. Maksudnya awak tengok itu sel dia, the pseudopodia, called as pseudopodia, will start to extend its pseudopodia, and surround it. Macam terperangkap dia lah, terperangkap ini partikel, pathogen atau food molecule. After that, it will engulf this and particle by endocytosis, what we call as phagocytosis lah. You need to use phagocytosis, and then later the vacuole will form. It will form the vacuole form, and then the lysosome will come fuse to this vacuole, and then it will hydrolyze this all of the particle. Hydrolyze this pathogen or food molecule, and then it will forms the particle, vesicle particle. So normally then it will release out lah. So this one is endocytosis. So normally phagocytosis is only involved until here lah. Normally is sampai sini tak stop lah, boleh stop. So you see here, normally the fold of plasma membrane, so the plasma membrane will fold, it will start to fold just surrounding, dia akan try to terbangkap ini molecule, food molecule or pathogen. After that, it will terbangkap dia, terbangkap dia, and then lysosome will start to fuse this vacuole, and release the hydrolytic enzyme to hydrolyze the pathogen or food molecule lah. All this lah. So this is what we call phagocytosis. But in pinocytosis, it will not extend the pseudopodia. It will not extend its plasma membrane. It will be trapped. Dia tak sama macam phagocytosis. Phagocytosis macam dia aktif punya. Is aktif punya. Dia boleh bergerak ke sini. Lepas tu dia nampak lah ada pathogen atau makanan, dia just terus pergi, and then it will extend, dia akan ambil makanan tu lah. Macam dia akan ambil makanan punya. But for this, it is not, dia tak akan ambil. It will be trapped. Kalau ada ion macam microvilli, one of the example, microvilli is our small intestine punya part, right? So dia tak akan bergerak punya, dia akan dekat sini saja. Kalau ada dissolved material, dissolved ion datang je, dia akan trap dia. So ini macam passive punya method. Dia tak akan bergerak punya, dia akan dekat sini saja. Kalau dia ada makanan datang, ada dissolved material datang, dia baru tutup dia. So it will pinch off into the cytosol as small fluid vesicle. Kalau dia ada mineral ion datang, dia akan close dia. So ini macam more passive method lah. It will not extend the plasma membrane one. It will tunggu je. Tunggu sampai ada particle datang, dia baru close. So once it's closed, it will form this pinocytotic vesicle. And then it will transfer to the cytoplasm lah, for the further modification. Okay, so this is what we process of pinocytosis. Okay, another one is about cell-mediated phagocytosis. Okay, ini special sikit, dia ada receptor punya. It involves the receptor on outside the cell. It will involve the receptor outside the cell. Maksudnya, dia akan very highly specific compared to phago and pino. Phago and pino dia tak kisah punya. Dia tengok je apa se. Macam phago dia tengok makanan atau pathogen dia makan je. Dia tak pilih punya. Tak pilih, saya makan ni, saya tak mau makan ni, tak apa. Dia makan semua je. Pino pun sama. Semua dissolved material yang boleh dia terperangkap, dia makan je. Tapi yang ni dia bukan semua benda makan. Dia ada satu receptor yang highly specific punya. Hanya the solute particle or ion that have the same shape with this binding site of receptor, dia baru boleh makan. So when for this diagram, you see that the purple one triangle solid particle is meshed with the receptor, and then it bind to the receptor. Once it bind, it will start to pinch off and then fuse bound together with the receptor, and the particle bound to it to become the coated vesicle. Become the coated vesicle that bound with the receptor. Although it already become receptor, but also still had the receptor one. So later it will go to the further function. So one of the example that you can use is LDL, low density lipoprotein, cholesterol. One of the cholesterol. yang ni cholesterol. Low density lipoprotein particle. Our transport of the cholesterol lah. Macam mana, how our cell and consume the cholesterol one. So this is the method. So it will use the cell-mediated phagocytosis, meaning that our plasma membrane will contain the LDL receptor. LDL receptor. So once the LDL particle, the cholesterol come to this, but come to this cell, because of it has the LDL receptor, it will bind to it. Once it bind to the receptor, and then it will start to will start the endocytosis process. Okay, it will start to pinch off the particle, and then it will start to form the coated vesicle. It will start to form the coated vesicle. Form the coated vesicle, and then after that it will form the uncoated vesicle. So the coated is because of the clubbing. Clubbing. Inilah clubbing. And then after that one endosome masuk, lepas tu ini proses dia lah. Ada endosome masuk, receptor are returned to plasma membrane and recycle. And then it will release, the receptor will be released out, will be released out because of the endosome, and then finally the vesicle containing LDL will fuse with a lysosome, and then lysosome will hydrolyze this chromosome, and then to produce the free cholesterol lah. Free cholesterol. And then this cholesterol will be used by the cell, either used by this cell, or it will undergo endocytosis to release to the other part of the cells. So this is the mechanism, or one of the example lah. One of the example of the cell-mediated phagocytosis. Yang paling penting awak kena tahu adalah dia ada satu receptor yang paling, ada satu receptor. So normally, so this one, the differences is highly specific lah, compared to the pinocytosis and phagocytosis. Okay, next we go to the osmosis. So osmosis is actually one type of the simple diffusion, but this only specific for water molecule only. When we have the movement of the water molecule in different concentration, then we will use the terms osmosis. So actually the definition is same as the simple diffusion, but the difference is it will involve water. Awak kena katakan water. So actually it is same as simple diffusion. So the water molecule will diffuse from high punya water concentration to punya low punya water concentration. Sama dengan simple diffusion. Okay, so this one I think you guys semua tahu kan macam mana tengok ni, yang ni famous kan. So you see that there are two different particle, right? One, the left hand side is low concentration, right hand side is high concentration punya sugar. So normally if you know that our simple diffusion, this one water contain more water, right? So the water will diffuse from left hand side to right hand side, because this one will contain more water. So in simple diffusion, it is down concentration gradient, so it will go to the right hand side. So after certain time, you will see that the right hand side punya tube sangat tinggi punya. The water content is more, because more water will be diffused through this membrane to the outside one. So inilah structure dia. Ini. Okay, so you see that water will move from area of high to lower free water concentration. Okay, so osmoregulation for the cell. So I think this one semua tahu kan? Hypertonic, isotonic, and hypertonic. What are they? What is hypertonic? So hypertonic is the solution that when your cell emerge in high water content, like distilled water. Isotonic is normal condition, half-half, 50% water, 50% solute. And hypertonic is that the condition which is high solute concentration one. So for animal cell, normally in isotonic E is normal condition lah, because the water in and water out is constant, is in equilibrium state, same as the plant cell. But plant cell in isotonic is not normal one, normal condition. Normal condition one. So what we call as flaccid. Plant is in flaccid condition. But and then when the cell is immersed in highly water contained, normally our animal cell will be burst lah. It will be burst because too much of water come into the cell. So our animal cell does not contain any vacuum. So when too much of water, dia akan pecah, memang akan pecah. Dia macam balloon lah. You can imagine balloon. Kalau balloon awak pump banyak air ke, banyak udara dekat balloon, memang dia akan pecah kan. Sama juga awak pump banyak water pun akan pecah kan. So same as the animal cell, because animal cell does not have cell wall and also does not have vacuum. But for the plant cell it will become turgid. So more water, plant cell bagus kerana plant cell ada vacuum nya. Dia ada vacuum. Dia have this very large central vacuum, right? So all of the water can store at there, and vacuum can be expand very large. So normally in hypotonic condition, it will become turgid. So when plant cell become turgid, it will become normal. It should be normal for plant cell. Kerana plant memang suka water compared to animal. But for hypertonic solution, so in hypertonic, if the animal cell is immersed in there, so water is out from the cell, but the hypertonic, the animal will not die. It will not easily die compared to hypertonic, but if too hypertonic also will die lah. But if hypertonic, the animal cell, the red cell, it will become shiver shape. And then for the plasma membrane, for the plant cell, if it immersed in hypertonic, more water contain keluar, dia akan become plasmolyzed. But the cell will also not die. But if it is too hypertonic, the plant will also will die lah. Because plant nak water punya. Kalau tak ada water, memang dia akan die punya lah. So this is what we call as plasmolyzed structure. Okay, next is the last topic lah. Yang ni paling penting punya calculation. This is the calculation topic. So, water, solute, and pressure potential. So, you need to know all of that definition. So, water potential definition, if you want to easy to understand, you can imagine that it is like the water concentration, water content. It is the physical property that predict the direction in which water will flow by measuring the free energy of the water. Dia macam you can imagine that it is like the water content lah. Because it can predict the direction of the water. It can predict the direction of the water when compared between the cells lah. And then solute potential is the potential of force of attraction toward molecule caused by dissolved solute inside the solution. So solute potential, you can say that it is like the solute concentration, because it is the force attraction to the water molecule caused by the solute. So normally solute is our solute concentration lah. Like sugar normally sugar lah. And pressure potential is the component of water potential due to the hydrostatic pressure that is exerted on the cell water in the cell. So these are the three definitions you need to memorize, and then these are the formula. If you want to calculate water potential equal to solute potential plus pressure potential. Ini formula dia. Okay, so normally this one is about the calculation question. Dia akan keluar soalan macam ni lah. Normally objective or structure question akan keluar punya. So this one I don't want to give answer. You can try yourself. Senang je, memang senang je. Yang ni adalah salah satu passing question. So maybe you already do it or not do it, up to you. So I don't want to give the answer. So you can try yourself, and then you're not very conform, you can ask in the group, and then can discuss. Maybe you can do it and then discuss. Okay, so these are the two question, example question. You can screenshot or whatever. I will also screenshot it into the group. Then you can you can make a poll. I will make a poll in the group, in the channel, and then you can answer it. Okay, so that's all for this chapter. Chapter 3. Sana kurang dia, memang sana short chapter. Already teach all in 1 hour 40 minutes. Wow, 1 hour actually within 2 hours I can finish this chapter already. So this chapter actually is, I can say, is the easiest chapter lah. Easiest chapter and short, easy and short compared to the chapter 1 and chapter 2. Chapter 1 and chapter 2 also easy but long, panjang. Tapi yang ni chapter easy but short. So hopefully can understand it. So any question? Okay, tak ada question. So hari ni ada satu question. So bagus. So any question or not? Okay, if no question, tomorrow I will proceed to chapter 5. Memang akan tak masuk chapter 5 lah. Kerana chapter 4, kerana chapter 3 slide ni saya pun siap hari ni juga. Chapter 4 memang tak dekat progress lagi. Hanya dekat 4.1, 4.2, 4.3, 4.4 belum lagi. So esok saya akan masuk chapter 5 lah. So chapter 5 cellular respiration. This chapter I think is semua punya minat lah. Semua minat dekat chapter 5 lah. Chapter 5 and chapter 6. So okay, so that's all. I think that's all for class today. Yeah, that's all for class today. So hopefully you guys can understand from class today. So you can try the question here. Okay, so that's all. Thank you for attending. Bye. Have a good have a good night or have a good rest. Tomorrow ada class kan, so have a good rest. Bye.